for the opportunity of presenting some of the work we're doing on tuberculosis at the moment. So, TB is a disease of poverty. It's mainly in poor parts of the world where there's the most TB, but over the last 20 years there's been an enormous progression in drug resistance. And so in the 1990s, multi-drug resistant tuberculosis emerged and is now most prevalent in India and China. And then in 2005, extensively drug-resistant TB emerged and rapidly spread around the world by 2008. And then most recently, totally drug-resistant TB has emerged. So there's no doubt that we're losing the battle against TB at the moment. And the mortality stays pretty fixed at about 1.5 million people per year. And why are we not making any progress? So we would argue that a key issue is that the fundamental paradigms that people are working from in the world of TB are incorrect. If you open a journal, you will see this life cycle presented. So TB is coughed by a patient with pulmonary disease, it's phagocytosed by a macrophage, the granuloma forms, and that breaks down and reinitiates the infectious cycle. And so that would be in all major journals. We've argued for a while that in actual fact, the initial implantation event is in the lung bases, and the reactivation event is in the apices. And so in actual fact, there are two immunological loci. And we need to understand those two events to really be able to address the TB pandemic. And I'm just going to summarise a little bit of the work we've done that originates from trying to address that. So this is a patient with TB. All 6 samples were positive for bacilli, and they'd been symptomatic in South London for about 6 months before they presented to healthcare. And so it's these patients who are the most highly infectious and drive transmission. And if you try and consider what is causing that lung destruction, it leads due to this family of enzymes, the matrix metalloproteinases. And to put it very simply, by studying samples from patients, you come to this enzyme, NAMRIP1, which is causing lung destruction. And this is the enzyme that the tadpole uses to degrade the collagen in its tail as it undergoes metamorphosis. So we know which enzyme is causing the lung destruction, but to really understand the process, we realised we needed to move from standard 2D cell culture to 3D cell culture. And you can see the display of the system outside, but essentially we realised at an early stage that you need cells in 3D to really investigate the host-pathogen interaction. And so our system relies on the fact that when you spray alginate into calcium chloride, you get these little microspheres. And so if we may have a syringe of alginate, we can put human cells, collagen, tuberculosis in, put it through the sprayer, and we can generate as many spheres as we want. And we have a multiplex readout. So in published work, what we've shown is essentially you need to incorporate collagen to mimic what's going on in patients. And so these are the cells immediately after being sprayed. We get these nice collections of cells forming after about 10 days, and we see multinucleate giant cells. And Liku has shown that the presence of collagen makes the cell survival better and controls the TB growth better. So in the context of antimicrobial resistance, it's very interesting to then ask what happens when you add antibiotics. And we can model the TB growth by having genetically modified TB which spontaneously produces light. And so the amount of light equates to the amount of TB, and there's the TB growing. If you put on your standard antibiotics, you kill the TB. A very important antibiotic against TB is pyrazinamide, but in normal cell culture it doesn't kill TB, there's a short dip down, whereas in the 3D system we can kill it. So it tells us that TB is becoming in an environment where it becomes antibiotic sensitive. And so we can identify new antibiotics which kill TB and shorten treatment. But that still is a poor system because the antibiotic concentration is fixed. And if we're really going to model events in vivo, we want to model what's happening in a patient where you take a tablet and the antibiotic concentration goes up and down. And again, Southampton's been an ideal place for this because we collaborated with Zunli, and he made us this beautiful microfluidic plate so we could irrigate through. And we made it and it didn't work. But it didn't work because there wasn't enough light, and with some optic amendments, so including putting a mirror underneath to reflect the light, we're now able to perform experiments, and we can model different antibiotic concentrations. And so you can see that we can kill the bacteria quicker by high peak concentrations, whereas if you're modelling someone who doesn't absorb their antibiotics, you get low peak concentrations. So we think this is a powerful system to model infection in vivo. A second spin-off is novel diagnostics. So as the enzymes break down the matrix, it releases these factors, P3MP from type 3 collagen. That's just one factor we've identified, and so we're collaborating with chemists, Spiro Garvis and Topher Welkers doing the bioinformatics. And by using his mass spec platform, we've identified this many upregulated and this many downregulated proteins, and that's just from the first segment, so that work is actively ongoing. And with the kind of near-patient assays which Rob Easton's developed, we think we can do population screening. And to model the effect of population screening, we've collaborated with Christine in mathematics, and you can see this is the Nigerian pandemic, what's happening without any screening. If you come out with a good screening test, you can control the TB pandemic rapidly. So, to summarise, what we're doing in Southampton is we're trying to identify new biomarkers to identify the people who've got TB. We have diagnostics and mathematical modelling to show what will happen when we do population screening, and the 3D cell culture system hopefully should identify new antibiotics in clinical trials. And I just want to thank all the people who are involved, but most importantly the patients who have taken part in the research. Thank you very much.